likely all sitting on land that was stolen from indigenous peoples who were forced into reservations during the Indian Removal Act of 1830. And as our speaker today gets to talking about some super interesting stuff, I'm sure you will all have some really great questions. Please feel free and don't forget to put those in the chat as you think of them, and we'll curate those and ask those at the end. So today I am super excited to introduce Dr. Bethany Hoye, who is currently at the University of Wollongong in Australia. She is a lecturer in the School of Earth, Atmospheric and Life Sciences, Faculty of Science in Medicine and Health, and Deputy Director of the Center for Sustainable Ecosystem Solutions. I hope I got all those right. Bethany got up early enough today before her young children even to do this talk, so I wasn't able to vet some of that information, so please feel free to correct me if I'm wrong, Bethany. So Bethany received her undergraduate degree from the University of Wollongong, her PhD at Utrecht in the Netherlands. Her first postdoc was at Deakin University, and Bethany and I met when she postdoced in the lab of Pete Johnson at the University of Colorado Boulder, where I was also a postdoc, and spent a lot of time in my office that was a broom closet. She didn't share the broom closet with me, but was in the broom closet / office next to mine. And as a postdoc, sometimes it can be a little isolating, and but I had the fantastic pleasure of working very closely with Bethany, who's a great colleague and friend to discuss science with, bounce ideas off of, and it was just a really fabulous experience. So I'm very excited to have her here today. Hey. In the lab of Pete Johnson, we studied malformations in amphibians. Bethany has since gone on to author numerous papers in disease ecology, including some on animal migration and disease. And she published a highly cited science paper linking bird migrations to biodiversity ecosystem function relationships. I thought she'd be a great speaker to our series because of the growing interest and expertise in disease ecology in our Tyson community, and her expertise in avian ecology, which is another growing interest here at Tyson, despite her focus on gulls, which is apparently painful for some ornithologists to listen to. But Bethany has recently begun work looking into gut microbiota in birds, which will be a focus of her talk today. So take it away, Bethany.
Sorry, just taking a while to unmute. Thanks very much for the intro, Kim. Most of what Kim said is true, although not necessarily as wonderful as she might make out. I guess I had it in my title, Scary Bits of Bugs, because I really wanted to emphasize, for those of you that are aware of any of the theory in disease ecology, how profoundly different antimicrobial resistance is to studying the transmission of any whole organism that's causing a disease in an animal population. And I was just going to start right at the beginning. Some might be really familiar with this information, but there are others of you who might not. So I just wanted to make sure everybody's on the same page in terms of what we're talking about. Where in the media you might hear about superbugs, like the one pictured on the right, but really what we're talking about is a bacteria that's changing its phenotype. So instead of being susceptible to the antibiotic, which is the image you see on the left, where around a disc that's been soaked in a clinically relevant concentration of an antibiotic, there's inhibition, that bacteria can't grow. Once a bacteria acquires a resistance gene that encodes mechanisms through which it can evade that antibiotic's action, it's then able to grow even in the presence of that antibiotic, which is the plate that you see on the right. And that tends to be an increasing problem, largely as a result of both overuse of antibiotics in populations that require them for treatment and misuse of them, so that they're then allowing opportunities for bacteria to escape or become more, populations to be more concentrated in resistant organisms. So but the data that best available, most widely available, is from Europe. And you can see in both of these plots, on the left, we have the bacteria Campylobacter, and on the right, Salmonella, and how frequently in production flocks resistance to a key antibiotic class, the fluoroquinins, is seen in Europe. The green bar represents the means of the number of flocks that are resistant, and you can see in Campylobacter, both in chickens and in turkeys, almost 70% of flocks have resistance to the number one antibiotic that would be used to treat that disease in production flocks, but up to 100% across the member nations of the EU. And likewise with Salmonella, we're seeing really, really high levels of resistance there. And that's not just a problem in production animals, also a really big problem in humans. So for the same two bacterial species or group strains, you'll see that over 60% of people who present with a Campylobacter infection, which tends to result in some form of irritable bowel syndrome or ongoing gastro, are then resistant to the number one antibiotic that would be used to treat that. Salmonella is much lower, only around 8 or 9%, but it's on the rise. Hopefully, you can then draw the conclusion that what's happening in livestock isn't separate from what's happening in humans. They're actually interlinked in a One Health sphere. And there isn't just one antibiotic. So I've listed 9 example antibiotic classes. So they're broad groups through which those antibiotics act on a bacteria. Some are able to disrupt a cell wall or disrupt a particular replication step for the bacteria. And that separates them into different classes here with the different colours. As with the original plate I had on the first slide, the one that's circled in green is representing the whole plate is covered in a bacteria that is susceptible to those with the inhibition ring around them. But you'll see that there are 4 different antibiotics here to which this particular bacterial strain is growing over the top of. So that means it's resistant to it. And they could be from a range of different antibiotic classes, which therefore represents a whole lot of issues when it comes to treatment. So you've got multiple different classes that bacteria is susceptible to, leaving very few treatment options. When we're thinking about how does that occur, it's not as if that one bacteria needs to, over the course of its lineage, be exposed to each of these 3 different antibiotics that I've got illustrated here. It's not as if in order to become the multi-drug resistant superbug that's over on the left, it has to have been exposed to both the blue, the red, and the yellow. And that's because bacteria can share their resistance genes and a whole lot of other what we call functional or accessory genes between one another when they cohabit. And so it's not necessary for them to be exposed to each antibiotic before they have resistance to it. And they even don't need to be exposed to any one of those if other lineages around them have been. So how does that happen at a molecular level? Well, those resistance genes are actually things that sit in what's called gene cassettes, generally represented as circular when they're on their own, and they can then be transferred between 2 bacteria in a bacterial community in one of 3 major ways. The first is bacterial transformation, which is illustrated here from a review from 2006. We'll see that those resistance genes are embedded within the chromosome of the bacteria. Some DNA is released and then is able to be absorbed in the recipient cell. You can also have bacterial transduction spreading in a similar way, where the resistance gene, which is indicated in the red on the left of the 2 bacteria, is picked up by a phage that's also infecting that bacterial cell. that phage is released, and as it moves to the next cell to try and invade it, it takes that resistance gene with it. And these are mechanisms for single gene transfer. But multiple genes can also accumulate along what's called mobile genetic elements. So on the far left here, we have these gene cassettes containing individual resistance genes, which have ATTC sites that they use to align on integrons. And once you have one on an integron, it's much easier for additional gene cassettes to be loaded onto that integron. Those integrons then sit within transposons, which then are capped either side by insertion sequences and allows them to insert into much more stable mobile elements called plasmids, which tend to form rings and be highly mobile between different bacterial cells. When bacteria come together, and through a process of conjugation, that entire plasmid ring containing all of the accessory genes that are loaded onto it are able to move from one bacterial cell to another. So this level of multi-drug resistance and the mechanisms through which it accumulates is really problematic for a number of reasons. The first of all, it's for populations who are relying on antibiotic treatments and then having not only reduced efficacy of the current treatments, but reduced treatment options. So they may have had a particular antibiotic that was widely used and there's resistance developed to it, but because there's resistance developed to that first one, the plasmids are then more likely to take on additional resistance genes, reducing any treatment options further down the line. How does that play out at a sort of public health level? Well, at the moment, they have estimated at a very, very minimum level, about 700,000 people die each year from infections that should have been able to be treated by antibiotics but aren't. In the next, up to 2050, this is predicted to increase to about 100 million people, largely because of that proliferation of multi-drug resistance, and so then the reduction in treatment options that are available. And that would ellipse some of our largest disease killers, both infectious and non-infectious globally. To put this in perspective, though, it's not going to be evenly distributed across the globe. As you can see, North America, Europe, and Oceania are least affected, and areas like Africa and Asia are seeing around 4 to 5 million additional deaths every year as a result of antimicrobial resistance. So this is a major area of public health concern, but it also is concerning that it isn't, the mechanisms allow antimicrobial resistance and multi-drug resistance to not be confined to the populations that are using those antimicrobials, and allows transmission into the environment and also into wildlife, because bacteria that are exposed to a bacteria that contain resistance may take it on through that process of conjugation. And wildlife is my main interest, where across, I've just put in some of the prominent examples of species that have seen antimicrobial resistance reported from them. Everything from penguins in the Antarctic through to foxes in the Arctic and everywhere in between. One of the groups that has repeatedly been cited as being very high in carriage of antimicrobial resistance has been the Laridae, or the gulls. As you can see, the silver gull that I'll talk about is on the top left. Underneath it is the black-headed gull, primarily reports from Europe and Central Europe. The yellow-legged gull reports from Portugal and Spain, up the top on the right, and then down below the glaucous gull, and most of that work's come out of Alaska. So it's globally distributed in species that tend to interact with humans in lots of different ways. Generally, when I'm talking to ecologists about antimicrobial resistance, most people's immediate reaction is, but why does it matter to wildlife? It's not as if they require antibiotics in order to overcome their infections. They would be dying from them anyway. So why should we worry about it, and why is it an ecological issue? Well, for a start, it's ecological in its process, and it's far more complex in its disease ecology than simple transmission. And so it requires ecological systems type thinking to look at those transmission processes and interventions that would reduce them. At the same time, there's potential for there to be linkages between resistance genes and virulence genes on those plasmids, where virulence genes are functional genes that code for how a bacteria is able to evade a host's immune response and a host's defense systems. And where they're thought to be functionally linked means that carriage of resistance could also mean that that bacteria brings with it increased virulence, increased pathogenicity in the host. The other side is back to the risk that animals pose for those populations that do rely on antibiotics, either as vectors, as kind of illustrated here, moving from one place to another. Like Kim said, I've worked quite a lot on migration in animals that are migrating thousands of kilometres, but there's potential for those antimicrobial resistance genes to be carried with animals when they're moving. There's also potential for their bacterial communities in their complexity and in their diversity, potentially amplifying drug resistance or changing what the resistance profile compared to the donor populations. And at the absolute very least, wildlife represent really good bioindicators of the pollution that we might be putting into the environment and the scale of the problem that's out there. So with that in mind, Andrew Ramey and his colleagues up in Alaska had put together a review a couple of years ago looking at what are the really key questions that need to be established in wildlife to better understand antimicrobial resistance. And this feeds into a much more holistic approach to antimicrobial resistance stewardship and finding interventions or strategies to reduce the risk of antimicrobial resistance and increase the longevity of our current treatments. And the first question really centres around what are the point sources? Where is this resistance coming from to be in wildlife in the first place? And there are a lot of correlational type statements made around exposure to human or livestock environments where antibiotics are used, antimicrobial resistance is high, and animals are then using those environments are thought to be higher in resistance. And while that's the case, it does still represent a bias in the way it's sampled. So there are a lot of studies of synanthropic species, and when those species are long distances from those point sources, they are also found to carry resistance. Secondly, those individuals that are at a particular resource have a huge variation across the population in their level of resistance. So it's not as if every gull that's in a town has high levels of resistance, just some of them do. And the third complicating factor that means we're not really clear on point sources is that a gull that might be operating around an urban centre is exposed to multiple of these sources. So understanding which ones are contributing the most is something that really needs to be worked on, because you can imagine that if it's largely wastewater treatment plants, as you see down on the bottom left, then strategies to cover those or reduce wildlife entering those might be really important. Whereas if it's the downstream outflow from those wastewater treatment plants, then that involves a whole lot better water filtration and processing before it goes out into the environment. It's probably a combination of all of those, but that's where we started our project. And here we're looking at some of the major questions beyond point sources as well, in terms of how are antimicrobial resistance genes perpetuated in wildlife populations? Do these wildlife, once they acquire them from a point source, then maintain them, modify them, and result in onward transmission? And I'm going to say that what I'm presenting today, even though it's the sort of built off 3 or 4 years of field study and follow-up lab work, it's very much the beginning of a story rather than a complete set of answers. As Kim said, this work is out of the University of Wollongong. Most people have never really heard of Wollongong, so I've given you a map here. We're just about an hour's drive south of Sydney, which is the major, the largest city in Australia and the capital city of the state, New South Wales. Here in Wollongong, when you look to the north, we have this really unique topographic situation where we're bordered to the east by open ocean and then rocky headlands interspersed with coastal beaches. And then to the west, which is on the left, by an escarpment, which is about 1,300 feet above sea level with a little rocky cliff cap. As you look back down towards the south, that escarpment's still there, but it moves inland, expanding the coastal plain in between. And what you can see in the distance, I don't know if you can see my cursor, but near the horizon, there's a little strip of land out from the point which is called Hill 60, and that's the island on which our field studies are based. This is part of Dharawal country, the traditional lands of the Dharawal or Tharawal people, and these islands are of large cultural significance to them for foraging, and there are a whole lot of cultural artefacts on the site. So because it's a key breeding site for a whole lot of seabirds, and because of its cultural significance, it's close to the public. From the shore, you can see a little sliver in the foreground, that's a rocky islet, and then out the back is what's called Big Island, and that's the where we're doing most of the field work that you'll see in this presentation. And the reason that we're working out there is because it's the largest breeding colony in Australia of the silver gull. You can see it from the map on the right, that breeding areas or year-round areas are illustrated in purple, non-breeding areas are illustrated in blue, and this coastal species has a really broad across the continent distribution. But the largest breeding colony, as I said, is right here, just 10 kilometres down the road from the campus. So we have an animal species, and we're looking at both the adults and the chicks of each year. And we have an environment that contains a whole lot of landfill, human waste, separated now into human unrecyclable waste and food waste that's going to large-scale compost, so they're at two separate sites. We have a whole lot of dairy cattle, which look a little different to these, and we have several wastewater treatment plants as well. And we also have some history of bacteria. So back in 2012, there was a study of 3 gull populations done by Monika Doljeska from the Czech Republic, and she'd found that the birds on Big Island actually had really high levels of salmonella infection, which has been linked to salmonellosis in the past. So we came into this study because the gull population is declining, and they were wondering whether these salmonellosis events might be responsible for some of the mortality in the population. In order to do our fieldwork, we want to be able to catch chicks multiple times throughout their development. So to do that, we set up these plots, which are 5 by 5 meters, thereabouts, at the beginning of the breeding season, around 20 to 30 marked nests in each plot. And that way we know the adults that are on those nests by capturing them and banding them. And then we can follow the progression of that nest from eggs hatching all the way through until those chicks fledge. And although we sample the adults once, these are the chicks that we're sampling from those first couple of days that they emerge in a nest cup, when they won't go anywhere or move at all, as you can see in the picture, all the way through until they're ready to fly and we can no longer catch them. So roughly weekly, depending on storm cycles and our ability to get out to the island, we're collecting metrics of these chicks. We're also collecting 2 cloacal swabs, one for bacterial culture and one for metagenomic sequencing independent of culture, and a blood sample so that we can go back and look at both antibodies to key pathogens that we're interested in, and, as I'll talk about at the very end, some stable isotope work to ascertain diet. These swabs are brought back to the lab, the one for bacterial culture, and split, put in medium and then medium split so that we can enrich for Salmonella and also grow the entire Enterobacteriaceae community. And you'll see a couple of different communities here where we're having up to 20 culturable species coming through from an individual swab. We'll keep that community, which we've done some metagenomic work on, but we're also specifically looking to isolate individual Salmonella strains. So that's taking a little loop from a clear colony, replating it to get a clean culture, and then doing some DNA extraction to complete whole genome sequencing. In terms of what that does for us and what advantage that gives us, instead of going in for a targeted PCR-based sequencing or assessment of what's there, where we might look for known resistance genes indicated with the little orange bars in the targeted sequencing on the right, by completing whole genome sequencing, we're getting a picture of what's happening across the entire genome, both coding regions and known coding regions. And with some additional steps, we're able to then assemble the genome of the bacteria and their accessory plasmids to have a look at what's located with what else. So for those of you not familiar with whole genome sequencing, we're taking that entire section of host DNA, and by host I mean the bacteria here. It's broken into small segments. Those small segments are then sequenced individually. And then you're using a whole lot of computational programs to align those, where you've got here, it illustrates there being 5 rows of a depth of sequencing depth of 5, but we're actually having each section of that fragmented DNA sequenced at least 60 times. So then you get a really high likelihood assembled sequence to work with. And our very first year, which was 2017, we found one isolate that was extensively drug resistant. First, we'd isolated this strain and taken it for that little disc diffusion phenotypic assessment at the local hospital, and they were alarmed that it was resistant to so many different classes of antibiotics, and that came up in the sequencing as well. So here I've got a phylogeny where you're not expected to read any of the tip names or any of the columns, but the tip names refer to all available strains on public databases for what's called Salmonella enterica serovar agona. So a particular strain of Salmonella that's an emerging pathogen in humans and in our agricultural sectors. You'll see a lot of the whole genome sequencing data that's available globally comes from the UK, indicated by the Union Jack flag. And our isolate is this one that's highlighted in yellow. And so it doesn't have any really close relatives, but it's clustering in with lots of things that are found internationally in the US and in Denmark. What's represented by the columns to the right are the resistance genes that it carries. And as you can see here, it's carrying a whole lot of resistance that isn't present in its closest relatives. And it carries 2 plasmids, which are indicated in the blue squares, that are completely different to what's carried by any other Salmonella agona that's currently available. What does that look like as a plasmid? And this one might do your head in a bit, but I'll try and walk you through what's where and what does it tell us. So the legend up on that top right is an indication of what the colours represent in the arrows in the outer ring. So the outer ring, with all of the arrows pointing in lots of different directions, is the HI2 plasmid, which is one of the 2 drug-resistant carrying plasmids that was in this SG17-135 isolate that we had. And the plasmid backbone is in green. Then down the bottom, you'll see that there's resistance region 1 and resistance region 2. In those resistance regions, which are spread out and illustrated better in the very bottom, you'll see that there are a whole lot of heavy metal resistance genes indicated in aqua and antibiotic resistance genes indicated in blue, and the insertion sequences that allows them to gain entry to the plasmid. And you can see that there are a whole lot of different resistance genes, but they are also have different flanking of those insertion sequences, looking like multiple acquisitions. The ring itself tells us how similar different parts of that plasmid are to, I think, 8 or 10 different plasmid sequences that were available online. And so they're listed on the right, and the shading just represents the percent nucleotide identity. So you can see that for the vast majority of the plasmid backbone, there is nearly complete homology with other published plasmids of this type. But when it comes to the resistance, that really breaks down, except for the one that's in the outermost purple, the CP33347, which is down the very bottom. And it's the outermost ring closest to our arrowed plasmid. And you can see that that one contains the heavy metal resistance genes and almost all of the antibiotic resistance genes that we had. And there are only a few little gaps in that outer purple ring, showing that there's very, very little difference between the plasmid that we have and what was published in the literature under this CP03347. And interestingly, that came from pork farms in China. So it's something that's separated by 5,000 or 6,000 kilometres and no known linkages between those two, but really high genetic similarity. So from this, we started a bit of a collaboration both with the local hospital and the local pathology provider to look at what's in not just gulls with reference to what's in genomic databases, but gulls in reference to the local human population, so that we could really get a time and site-specific degree of similarity between these two populations. And we had the historical data from 2012 from the Czech Republic group that had worked on silver gulls. And then we've done 4 subsequent sampling years here. And 2019 and 2020 are grayed out because although we have some of the gull data there, I won't present it today because we're a little COVID delayed on some of the hospital components. And by comparing between years, we're able to look at what's being perpetuated in a gull population in terms of the bacteria themselves, the salmonella, and in terms of the resistance genes they carry. By comparing over time in the human population, we're also able to look at perpetuation in that population. And by comparing between the two populations in a given year and across years, we can get a much better idea about how much transmission might be happening between these two populations. And like I said, we're also working on sampling those chicks multiple times, up to 6 times across their development. And so then we have at the individual level some idea of perpetuation in individuals, whether an individual's maintaining its resistance genes and a particular strain of bacteria, or whether there's high level of turnover. So here's a simplified phylogenetic tree representing the relationships between just salmonella isolates that have come from Wollongong. Everything that is in a shade of blue is from gulls. Everything that's in a shade of orange has been isolated from humans. And they're clustered into sequence types which roughly correlate to serovars, so large differences in the strain itself of the salmonella. And we're going to have a look at a couple of these in detail to get a sense of what is going on. So we'll zoom in here on the Typhimurium clade. And when we look at it in isolation and build a tree based on SNP distances, you can see, although there's a large number of gull isolates that all cluster together, and that's that blue section and the bottom of that panel, where the ones in blue are original sequences and the ones blocked out in white are where we've taken multiple colonies from that single plate, because we wanted to see whether an individual was at any given point in time infected by multiple strains of Salmonella, or whether it just had single strain of Salmonella infection. And at the moment, we're only seeing that they're ever colonized by a single strain of Salmonella. But up the top, you can see that there are sister clades between the hospital in light orange and a gull, and we see that repeated across the tree. And when we've put in the 2019 and 2020 data, we're seeing that multiple times. We have up to 50 instances of very, very little SNP distance between the bacteria that's found in the human population and the bacteria that we're seeing in the gull population. Just as another example, we zoom in here on sequence type 19, and you see the same thing where you've got human infections nestled among gull infections and vice versa. When we come to have a look at what do those bacteria contain in terms of resistance, everything contains aminoglycoside resistance, but the rest contains very little additional resistance. That is spread across 41 different resistance genes to 12 different antimicrobial classes. And each different class is listed there in the rainbow colours to help us separate them when I get to a much more complex figure. So between the two populations, we see that there are more unique resistance genes and more genes in total in the gull population. But the number of individual bacterial isolates that have resistance to more than one antibiotic is more than twice as high in the human population. That's complete counter to, once it's multi-drug resistant, how many resistance genes are in a multi-drug resistant isolate. So in gulls, there tend to be more resistance genes on average once it is a multi-drug resistant strain, even though those multi-drug resistant strains are seen less often. Overall, what that really tells us is what we'll have a look at here in this giant phylogeny, where the phylogeny is represented on the left, and it's just coloured so that you can remember which is gulls and which is humans. And the resistance heat map occurs for each gene as you move to the right. What you see is that aminoglycoside resistance in the purple occurs in everything, but resistance to other things is quite patchy. And the only ones that really are showing that resistance to multiple genes, so lots of different colours along their line, are the gull isolates. There are a couple of human ones, but there isn't correspondence between sister bacterial clades and the resistance they carry. So even though there might be indication of very, very recent transmission at the bacterial genomic level, there's been some shift in what resistance genes are in that bacteria. So to summarise what we've seen in terms of Salmonella in silver gulls so far, we have evidence of extensive transmission between humans and gulls. But like I said, they're distinct in their antimicrobial resistance gene carriage, and those genes are loaded on distinct plasmids. So something happens between those bacteria being transmitted and the incorporation of resistance genes. And it's a question that we're working towards understanding much more broadly in the bacterial community inside the gull. What I haven't shown here, because it gets a bit complicated for the figures and the time we have, is perpetuation. But across years, we're not seeing the same Salmonella sequence types in the gull population or the human population. And we're definitely not seeing perpetuation of resistance genes one year to another. Things that we're finding in different years as extensive drug resistance aren't then showing up again year after year. And I just had an honours student complete a metagenomics project where she's looked at the entire bacterial community that we could culture from a gull at multiple points across its development. And with that, you get an idea of all of the resistance genes that are in there in any bacteria. And what she's seeing is while there's lots of resistance in a chick at any point in time, between weeks, the presence of resistance genes is changing. And so across all of these, it really seems like gulls are acting primarily as a bioindicator of a whole lot of transmission events and a whole lot of environmental availability of antibiotic resistance, but may not be responsible for a whole lot of maintenance or the presence of a reservoir. Then we go back to my other passion, which is understanding how much animals move and how they use the landscape. And we wanted to understand where might be really high risk areas for these transmission events between humans and gulls. When might that take place? Because understanding those two processes allows us to start thinking about interventions that might stem transmission between the two populations. So first pass, we really needed to understand how are these chicks, which are confined to the island and they can't yet fly because we can still catch them, getting exposed to human pathogens that only occur off the island because the island's a closed reserve. While there's a shed there, it's just 2 shipping containers that contain our sampling gear and that we can sleep in if we need to stay overnight. But there are no facilities, there is no waste disposal, there is no toileting of any kind. So we're looking then at the adults, which are going to the mainland to gather food and bringing it back to the chicks to understand better how those adults behave and use the landscape. In 2019 and 2020, we attached GPS loggers to 38 different adult birds. And this is an honours project of Nathan Brown, illustrated here. And I'm just going to present some of the work from 2019 breeding season. So these loggers are getting positions every minute to 5 minutes. Once the bird leaves the island, that frequency goes up. And individuals vary a lot. So on the left, you have a bird with the logger ID 2492, and you can see it hardly moved in the entirety of the breeding season, 8 or 9 weeks. It's only using about 10 kilometres strip of space in the landscape. Whereas the bird on the right, 2496, is taking extensive trips all the way up to Sydney and back and doing a lot of marine foraging. So to classify that, Nathan went through and took birds for every minute that they're off the island, where are they spending their time, to have a look at proportional use of different habitats. And he's classified the landscape using GIS layers. And this diagram is just an indicator to get you thinking, get your eye in for what's going to be coming in a much more complex one, where you'll see this individual uses a whole lot of urban down the very bottom, spends a bit of time at the sewage treatment works, 10 to 20% of its time, a bit over 20% of its time at the landfill, and then a whole lot of increasing amounts of time across the breeding season out in the open ocean. So when we look at across those 30 individuals that we tracked in 2019, you can see that the number one outcome is that they're really, really variable. Some individuals are spending a whole lot of time in the marine realm, like 2456 up on the top left, and others spend a whole lot of time at the sewage treatment works, 3 rows down at 2499, and everything in between. All of the individuals are visiting some high-risk environment at some time, whether that's a landfill site or a sewage treatment site, and they're foraging while they're there. Nathan got to go and do a whole lot of behavioural observations at these sites, and at each of the places where we're seeing gulls spend lots of time, they were foraging. And although we could categorise them into broad foraging categories statistically, they that was across the season. You'll see if you zoom in on just 2457 up here highlighted in the yellow as an example, that changes from 1 week to the next. So it goes from using the landfill a little to spending a whole lot more time in bays and estuaries. And that's a dynamic that we're seeing across all individuals. They also allowed us to get some understanding of where individuals go when they're not breeding, which is really unique because although migration research is really well established in Europe and North America, in Australia it's thought that very few species undertake long distance migrations, and the human population is so different in its density and its layout that ringing studies haven't established predictable migrations. And our gulls are kind of similar to that, where the arrows represent they all just went to different non-breeding sites and spent time in their own little bay or estuary up and down the coast. Some of those were really close by. So where it says Port Kembla on that map, it's where the gull breeding colony is. This bird moved down to the bottom corner of Lake Illawarra, all of about 10 kilometres away, and sat there for about 6 months, just in one corner of a lake before coming back to the breeding colony. Whereas there are others, all of them, like you can see with this one, it's no longer foraging at the tip. And all of them moved away from using that landfill site, which is the complete opposite of what's been seen in Northern Hemisphere gulls, where they're using natural foraging for their breeding, and then they go back to anthropogenic foraging. Outside of that, we're seeing a complete reverse. We also had one individual that made some really long distance migrations. It actually took 2.5 trips to Melbourne before it came back to breed in Wollongong via a whole lot of inland areas. And when you add that up, it's the equivalent of flying something like Montreal to Miami and back. So to some of the GPS track, we're seeing that almost all individuals use some high-risk sites, but they use it to different degrees and at different times. What we also see is with that level of variability and with salmonella infections being only at about 5% of the population, that you're not able to match those two up. We can't do epidemiological investigations on that many individuals' movements. We're really going to need a whole lot more, either to track thousands of adults, which is untenable, or a different way to get at what's been fed to those chicks. So that brings me to some of what we're doing now, where our chicks have been regurgitating their food parcel as we collect them. And this one's a really mixed sample, so it's not particularly useful for developing an isotopic map. But it does show fragments of glass and plastic that we're getting, that there is a whole lot of anthropogenic food in there. But there are also some that are really a single prey item at a time, like these, what are called silverfish, tiny little bait fish, that we're then able to get the composition of the stable isotopes of carbon and nitrogen in this food source and come back and map the chicks' blood stable isotope concentrations to that to figure out diet sourcing for chicks. And it's revealed that they're eating some really strange things that we hadn't expected, like these moths and beetles and a whole lot of other invertebrates as well. And we're pairing that with a whole lot of selective plating across all of our communities that we've taken from the gulls, so that we can get a really good idea of resistance carriage independent of which bacterial species it might be in. And that will also feed into some work that we're doing, looking at the resistance sharing between things like Salmonella and E. coli and Klebsiella and other bacteria that are all occurring in the gulls, and we're seeing them quite frequently and really associated with human disease in lots of places. So to make sure that we leave some time for questions, I was just going to say I've got projects that if anyone's interested in, also on flying foxes, and similar questions there with changes to diet, changes to habitat, and microbial compositions that come with it. So if you've got any questions about those, you can jump onto either of those contact links. And I'd really like to thank, this is the work that's been done with a huge number of collaborators. It's not all my own. Down the bottom, there's Danielle, Maddie, and Nathan have all been honours students over the last few years, but collaborations with the local health district, with the Centre for Genomic Epidemiological Microbiology, and partners in land management as well. With that, I'd like to say thanks very much for listening. If you have any questions, for all ears.
Fabulous. Really, really great talk, Bethany. Certainly something that I hadn't thought about much before, and I'll be thinking about more in the future. And certainly, I think much of our community here hasn't heard much about this sort of work, and so thanks for filling in some gaps and helping us think a little more broadly. We have a few questions coming in. I would like to ask one to get us started. Is anyone else doing this work elsewhere? I mean, is this a pioneer in looking at this link between gulls and humans?
So the folks in Alaska have this Andrew Ramey and his crew have been working on Alaska landfill sites and non-landfill sites and sampling gulls there. And they've probably for nearly a decade, and they've just started doing some GPS tracking of their movements. And there's a little work out of Spain on some gulls as well. But in terms of understanding what's in wildlife more generally, it's really overlooked. So I was talking to one of our collaborators is a veterinary pathologist, and at his wildlife health sanctuary, they have a lot of koalas come in from like incidents on people, like roadkill, but not incidents, I guess you could say. And when they come in for rehabilitation, they're doing some sampling, and they're finding that koalas and wombats and rock wallabies and everything that's brought in is carrying high levels of resistance. They think primarily from interacting with livestock feces in paddocks and things like that. But it's definitely a whole lot more widespread than we previously thought.
Yeah, it seems like it's an interesting area of inquiry for a lot of other species that interact closely with human populations, like, rats in urban areas. And
I had a student try and do like some research on what uses landfill, for instance, and everything visible, like ibis and gulls and ravens and foxes, in the literature, but the rodent use of landfill is completely out of the literature, and we have really little understanding of how much interactions in that space.
Mm-hmm. Yeah, really interesting. Okay, here we have some questions from our audience. When you say you work with hospitals, does this include public messaging so the community knows what you are finding?
Eventually, yes. So at the moment, we're working with the hospital as their pathology unit is really interested in having a more holistic approach, a One Health approach to how they deal with their antimicrobial stewardship. And so they are very happy to share their isolates and help us look at that phenotypic susceptibility testing, so that as we develop a more broad picture, they have a program not directly to the public yet, because you need to have really clear outcomes before that comes through, but with GPs. And so we have an organisation with local GPs where we discuss research findings and implications and make planning for not planned, kind of data-informed choices about antimicrobial prescriptions for patients based on what resistance is around and what would be optimal to prescribe.
Mm-hmm. Sidebar, I'd like to talk to you further about that collaboration with those physicians. So a follow-up question from me, it seems like the physicians at the hospital that you're working at have embraced the One Health approach, which is fantastic. Do you have any sense for how widespread that acceptance may be, or you think we still have a long way to go?
I think it's probably beyond a lot of their horizons, not through any fault of anyone's, but just because of what's in their day and what they're dealing with. And it's not, I guess, not well embedded in our training of medical professionals yet. It's sort of put up as, they might even have a subject or a lecture on it, but it's not embedded in the way they approach medicine, I guess.
Yeah.
But there are glimmers of hope.
Right.
They're interested individuals that are willing to then bring others along with them.
Yeah. That's encouraging. Okay, we have another question from our audience. What do we know about gulls or other wild birds highly associated with humans and other pathogens that we may share with them? Do they also pick up our viruses, for example?
Yes. So they pick up our viruses, and gulls do a number of RNA viruses that might be zoonotic to humans. I haven't done a whole lot of virological transcriptomics because those viruses occur at really low frequency. So in order to pick them up, I did some influenza surveillance just because it's what my background research has been in. And they're definitely carrying some influenza, but where it's coming from and how frequently, it's, yeah, a can of worms to be opened.
You plan on investigating this in birds that migrate long distances? I mean, I know
That's a funny thing about Australian birds is that there aren't that many well-documented long-distance migrants. I've worked with shorebirds that come from, they breed up the very top of Siberia, and they come down the coast through the Korean Peninsula and the South China Sea and back to Australia. And in those populations, in particular, we see a whole lot of like influenza transmission between China and Australia, and evidence of exposure to a broad range of things. And in those, I'd really like to look at the antimicrobial resistance. But things like the gulls are probably undertaking really long distance movements, just not all together as a group, and hence migration. Yeah.
Got it. And a really good question here from Andres. What is your favorite gull?
We only really have one, so that's pretty easy. No, I mean, there's several gulls. They're a nice animal to work with. They obviously extremely intelligent, and I think all gulls are extremely intelligent. You wouldn't have something that's able to be so adaptable if it wasn't. So that's kind of nice.
I think maybe he might have meant which individual gull.
Oh, the really funny thing is,
I don't think so, but yeah.
Yeah, I don't have a favorite pet gull.
Okay, it looks like that's all from the crowd. This was really interesting. Thanks, Bethany, for getting up so early and talking to us about this really cool research. I look forward to talking to you about it more. But for now, we will say goodbye to our crowd.
Thanks very much, Kim. And if anyone's interested in doing research in Australia, we do have opportunities for both masters and PhD positions that are funded. So if you wanted to get in touch by Kim or whatever, looking for a research project, then get in touch.
Now it's clear why you were showing those really beautiful pictures of the area, just to show the landscape and how that affects the work. It's to show how beautiful this area is. So
it's reasonably enjoyable field work. Yes.
Fantastic. Thanks, Bethany. We'll see you.
Thanks, Kit.